Hello children, we were studying plant physiology, structure and germination of seeds. We started with generalized structure of seed followed by types of seeds on the basis of number of cotyledons and presence or absence of endosperm. Now today we will start with a detailed study of structure of a bean seed. And bean seed is an example of DICOTYLEDONOUS EXALBUMINOUS seed. That means it has two cotyledons and it is without endosperm. Children, you all have observed kidney beans. You all have seen it. Externally, the features are very simple, easy to understand. Now, just like any other seed, it is having a seed coat. The outer layer is a thick one. It is a smooth colored protective layer called testa, and the inner layer is a thin white membranous layer called tegmen. Then when we were studying the generalized structure of a seed, I told you that there are two more important things to observe, that is hilum and micropyle. So here in case of bean seeds also you will find hilum, that is a scar which shows that how the seed was connected with a stalk. It is present on the concave side of the seed. And micropyle, again very close to the hilum, you will find one small opening, one small pore called micropyle. And you already know that what is the function of micropyle. Its function is to absorb water and it helps in exchange of gases. Apart from this, there is one more structure called raphe. It is a ridge-like structure that is present in the median groove. Now, median groove means longitudinal depression, that is on the back side. A longitudinal depression is a groove. If you remove the outer covering, that is the seed coat, then inside you will find the embryo. And embryo consists of two parts, cotyledons and embryo axis. Now cotyledon, here it is without endosperm, so you all know that cotyledon is responsible for providing nourishment to the developing embryo. So, and it is dicot, so it is two in number. Cotyledons are fleshy and they are meant for storage of food. They provide nourishment. Then embryo axis, it is further differentiated into distinct regions. We will take up each one by one. The first part is mesocotyle, means that portion from where the cotyledons are attached with the embryo axis. Then epicotyle, it is the portion between the mesocotyle and the plumule. Then hypocotyle, the portion between mesocotyle and radicle. What is plumule? It is the leafy end of the embryo that will develop into shoot system. And radicle, it is the pointed lower end that develops into root system. Your children, you will see the structure. The lateral view of a kidney bean. Hilum and micropyle, you can easily identify them. The seed coat, the outer covering, that is colored, okay, it is generally reddish in color. So the outer covering, that is your testa, that is a colored protective layer easily observable, and hilum and micropyle, these things externally we can easily identify. Now if you soak it in water and you try to open it up, then inside you will be able to notice the cotyledons, plumule, radicle, and epicotyle and hypocotyle regions are also easily observable. Alright. Moving on to an example of monocotyledonous albuminous seed. Now monocotyledonous albuminous means it is having one cotyledon and with endosperm. And the example that we will study is of maize seed. Now you all know, you all have seen, you've eaten it also several times. Maize is a one-seeded fruit, and what do we call one-seeded fruits? They are known as caryopsis. The special characteristic is that the fruit wall is fused with the seed coat. So, if you observe a maize grain, the first thing that we can see is that the fruit wall or the pericarp is fused with the seed coat. One maize grain, it is almost triangular, and externally we can differentiate it into two parts. The upper part or the upper area is larger one, and the lower area is a smaller one. So you can differentiate it into two regions, the upper larger region and lower smaller region. The upper region is yellowish in color, and the smaller lower area, that is white in color. So the upper region is the endosperm, and the lower white smaller region is embryo. Alright, so externally maize is differentiated into two unequal areas. Upper larger part is the endosperm, the lower smaller part is the embryo. See children, the external appearance of maize. Then the next structure shows you the longitudinal section as well. Now, as I told you, externally we will find the fused seed coat and pericarp. Then hilum and micropyle. Now in case of maize, hilum and micropyle, they are not visible, okay, they are invisible. Then endosperm, since it is covering the maximum area, I told you that the large upper area is endosperm, so it covers the bulk of the grain. And endosperm, the purpose of endosperm is to store food. Alright, it is storing food material, reserve food material in the form of starch, and it provides nourishment to the growing embryo. One more speciality is there. It is surrounded by one cell thick layer called albumin layer. Now children, this albumin layer, if you see chemically, it is very rich in proteins, okay. It is a protein rich single celled layer. Its function is to nourish the embryo, plus at the time of germination it is also responsible for releasing some enzymes. So this albumin layer nourishes the embryo, plus it helps in seed germination. Then coming on to the embryo section. Okay, so we discussed the upper part of the maize grain, the endosperm and the albumin layer. Now moving on to the lower smaller whitish area, that is the embryo. Now here you see it has scutellum. It's written scutellum. What is scutellum? It is nothing but the cotyledon. Okay, the maize grain, it consists of a single papery cotyledon called scutellum. Now it does not contain any food. All the nourishment is done by the endosperm, so it does not contain any food. Alright, it serves to absorb nutrients from the endosperm. Then there is plumule. You all know what is plumule. It is the it consists of the growing tip of the shoot, and here in case of plumule it is protected by a covering called coleoptile. Similarly, radicle, that is the structure that will in future give rise to the root system. Here radicle is also covered with a protective covering, a protective sheath called coleorhiza. So here in case of maize, plumule and radicle, they are covered with a protective sheath. We did not find this in bean seed, right? So if asked that what are the common differences that you can observe in case of bean seed and maize seed. So what are the differences that will come to your mind? First thing is that in case of bean, the seed coat is thin and it is having a characteristic color. In case of maize, the seed coat is thin, but one speciality that the pericarp is fused with the seed coat. In case of bean, the hilum and micropyle are visible, whereas in case of maize grain, they are invisible. Bean seed, an example of DICOTYLEDONOUS EXALBUMINOUS seed, so it is having 2 cotyledons and it is without endosperm. Maize, here a thin cotyledon is present with a different function and it is only 1 in number, right? Then in case of beans, endosperm is absent. Maize, the nourishment to the embryo is given by the endosperm. Then in case of bean, the plumule and radicle, they are naked, they are not covered by any protective sheath, whereas in case of maize, they are covered by protective sheaths called coleoptile and coleorhiza. Then the reserve food material. In case of bean, the reserve food material is generally carbohydrates and proteins, whereas in case of maize, the reserve food material is starch. Alright. Now children, we have discussed all this, the internal structure of maize seed, right? Moving on to seed dormancy. Now what is seed dormancy? Dormant means anything which is not active, anything in its inactive state. So when do we say that a seed is dormant? Seeds which do not germinate even when it is provided with all the suitable conditions necessary for germination. So whenever a seed, even if it is provided with all the necessary conditions, it fails to germinate, we say that it is a dormant seed, and this phenomenon is called dormancy. Now there are several factors, there are different causes for seed dormancy. All right. Sometimes inadequate light or some internal factors may be responsible for seed dormancy. Now what is germination? All the processes by which this dormant embryo of the seed grows out and establishes itself as a seedling. So all the processes that are contributing towards conversion of this dormant embryo into a seedling is called germination. So it is not one, but it is a combination of all the processes. Okay, the entire activation from a dormant embryo to a seedling, that entire process comes under germination. Under suitable conditions, embryo overcomes dormancy, it becomes active and starts germinating. Now there are some basic conditions. Those conditions must be fulfilled for germination to happen. What are they? Water, light, oxygen and temperature. Alright. We will discuss these conditions in detail, plus there are 2 ways of germination that we have to discuss, and there are some experiments related to germination. So we will be finishing those topics in the next session. Till then you have to revise till the topic that I have covered today. Alright children, thank you very much.